Hello, everyone, and welcome to today's EBD Group Bioeconomy webcast titled Diverse Approaches to New Chemical Modalities, Challenging the Undruggable Targets Beyond Conventional Small Molecule Drug Discovery, sponsored by Axcelead. This is part one of our three-part series titled Transform Your Drug Discovery, Faster Paths to Clinical Success. First, I'll cover some quick housekeeping items. If you experience difficulties with audio or advancing slides, refresh your screen with F5. If you are experiencing other issues, hit the question mark button to receive assistance. At any time during the presentations, you can submit your questions into the Q&A window on the left-hand side of your screen. And in 24 hours, you receive a link to watch the recording of this session. Let's now begin by introducing our speaker for today's session. We have Yoshito Terao, PhD. He's the head of the Chemistry Business Unit for Axcelead Drug Discovery Partners Incorporated. Thank you very much, Yoshito, for joining us today. I'll now hand it over to you to begin the session.
Okay, hello everyone, and thank you very much for joining today's webinar. My name is Yoshito Terao, and I am the head of Chemistry Business Unit at Axcelead Drug Discovery Partners. I've started a drug discovery career as a medicinal chemist in Takeda, and I have over 20 years of experience in drug discovery as a scientist, leader, and manager at Takeda and Axcelead. And prior to that, I received a PhD from Osaka University in Japan. I am passionate about finding new ways to create better medicines for patients. Today, I'm very excited to be here to talk about diverse approaches to new chemical modalities. This is a topic that is very important for our industry today. As we all know, many disease targets that were once considered undruggable are now becoming possible to address with new approaches. At Axcelead, we have been working hard to develop these approaches, and I want to share our experience and capabilities with you today. Today's presentation will show you how we can work together, and I will try to keep my talk simple and clear so it can be followed easily by both scientific and business audiences. Here is the outline of my talk today. First, I will briefly introduce Axcelead and our company background. And second, I will explain rationale for new chemical modalities that will help us to understand why diverse approaches are needed. Third, I will discuss the challenges of new modalities and how Axcelead addresses them. Fourth, I will describe our capabilities in each modalities: peptide, oligonucleotide, drug conjugates, targeted protein degraders, and RNA-targeted small molecules. Finally, I will give a short summary. Okay, let's begin with our company overview. We actually were created as a spin-off from two major pharmaceutical companies, Takeda and Teijin. This gives us something very special. That means that we inherited not only pharma experienced scientists, but also high-quality assets from these pharmaceutical companies, which makes us unique as a drug discovery CRO. And we have two sites in Japan. Shonan site comes from Takeda, and Neurohino site is a joint venture with Teijin. And as shown on the right, we now have more than 300 scientists, many with pharmaceutical industry experience. And our compound library is over 1.2 million molecules, which utilize hit identification, and we have more than 1,000 drug discovery project data. This is not just numbers, this is real pharmaceutical asset experience and knowledge we have inherited. And our recent business track record shows our quality. Now we have more than 290 clients with 90% repeat rate. And this high repeat rate shows that our clients trust our work. And we also have a research collaboration with companies like Lilly and Acadia. And most importantly, we have an 84% success rate for integrated drug discovery of lead generation and optimizations. This strong foundation allows us to tackle the challenging work of new chemical modalities that I described today. And this slide shows our platform. Our mission is to contribute to client drug discovery challenges with a pharma-grade platform. Please look at the left side. We provide a comprehensive integrated drug discovery services. This means our scientists support you from target identification and lead optimization to IND-enabling studies by seamless manners with pharma standard knowledge and data. In addition, to boost innovation in the right side, we combine the legacy pharmaceutical data with AI to improve drug discovery success rate. And we also expand beyond traditional small molecules into modalities such as peptide, oligonucleotide, which I talked today. In addition, we strengthened our capabilities through the strategic partners to adopt new technologies. And in short, we integrate people, technology, and data to accelerate drug discovery for our client needs. Okay, now let's move on to today's topic, rationale for new chemical modality and challenges and solutions in Axcelead. Why do we need a diverse approach to new chemical modalities? This question is at the heart of modern drug discovery. And of course, conventional small molecules are powerful, and they can act on intracellular targets. They are often orally available, and they are relatively inexpensive to manufacture. However, they also have limitations. For example, they are not always suitable for very large binding sites and not suitable for direct actions on nucleic acid and not suitable for cases where extremely high specificity or long duration are required. This is where new chemical modalities come in. As shown on the right side, the approaches can overcome the traditional limitations. As shown here, new modalities may address targets with wide binding site. They may directly act on the nucleic acid target, and they can achieve a high specificity and provide a long duration of actions. And why diverse approaches are needed? This is another question to new chemical modalities. And this is because different drug modalities can complement each other, and they provide multiple ways to treat the same disease pathway. And they also allow different administration route or injection or even one-time gene therapy, depending on the patient needs. In addition, they open options combination therapies. Let me give one example of SMA at the bottom. This disease now has a treatment of 3 different modalities: an antisense oligonucleotide, and gene therapy, and a small molecule. Each works in a different way, and together they provide a broader treatment options for the patient. And this example shows that why multimodalities are so important. I'm sure that future of drug discovery is not about choosing one modality over another. It's about having multiple tools and using the appropriate tool for each specific situation, I think. Of course, these new modalities come with the challenges. Now let me explain the main challenges we face with and how Axcelead provides solutions. First, their chemistry is complex. It requires advanced synthesis and purification technologies. Second, each modality needs its own evaluation system, different assay system, pharmacology models, and safety evaluations. Third, we need to decide the best modality for each target, which is not always obvious. At Axcelead, we have built solutions for all 3 challenges, such as advanced chemistry across modalities, a total evaluation platform, and experience to propose the right modality for each project. Let me show you our solution on the following slides. The first one is how we solve the complex chemistry challenges. At Axcelead, we have a full chemistry capability for candidate generation in drug discovery. Our all-in-one medicinal chemistry enables the drug discovery of a small molecule, peptide, drug conjugate, and oligonucleotide modality from hit to candidate. And for compound design, we use computational chemistry and data science and AI approaches. And for synthesis, we have capability for practical synthetic route design, scale up, and high throughput parallel synthesis. And what makes this special is that we can handle all these modalities toward a candidate by the highly experienced scientists. While most companies specialize in one area, but we can work across different modalities and even combine them when needed. This integrated chemistry approach brings more innovative solutions that might not be possible if you work with separate chemistry companies. And we also actively expand our chemistry capability through the strategic collaboration with external partners. For oligonucleotide, we have a partnership with the Nissan Chemical Corporation announced in the July 2025. This partnership will bring together Nissan Chemical technologies like MC modification amidite with Axcelead drug discovery platform. For antibody drug conjugate, we have a three-way collaboration with Lotte Biologics and Canaf Therapeutics announced in the June 2025. This collaboration focuses on the developing ADC toolbox like novel linker and payload. These partnerships show that we are continuously improving our capability and staying at the forefront of the new technologies. We believe that the best solution comes from combining the strengths of different companies. And the second challenge and solution is evaluation systems. As you know that for each modality, we need specialized assay and platforms. At Axcelead, we offer the comprehensive integrated evaluation system that covers screening, pharmacology, DMPK, and safety. As shown in the right side, we can handle many areas such as oncology, CNS, immunology, and cardiovascular and metabolic diseases across modalities. This one-stop evaluation platform reduces the turnaround time and help us to make decisions faster and with greater confidence. Let me give you a concrete example of how we seamless evaluation works. This is from the GPCR project. Look at this table. It shows how we organize the workflow from synthesis through to the in vivo efficacy testing. We conduct a continuous research across chemistry, screening, DMPK, safety, and pharmacologies because all function in the same building. In addition, all admitox profiling can be completed in normally just 1 week. This our speed advantage means faster decision making, more efficient use of resources, and ultimately faster progression towards a clinical candidate. And now let me explain the third one, how we approach the challenge of selecting the optimal modality for each project. I'm sure that the key is synergy between the client specialty and Axcelead platform. Client bring their specialty in in therapeutic area, proprietary technology, and target product profiles. And Axcelead bring our total platform technologies covering chemical modalities and our data and experience from the over 1,000 projects, which allows us to propose a diverse modality approaches. For example, if a small molecule does not work, we may move to the peptide. And if a peptide alone is not sufficient, we may consider a drug conjugate. By keeping this kind of flexibility, we increase the chance to create variable compounds. All right. Now I want to show you our specific capability for 5 new chemical modalities. This is the technical part of our presentation, where you will see the what Axcelead can do for each type of modalities. Okay, let's start with peptide drug discovery. Peptides are very important new modalities because they can address targets with wide binding sites that are difficult for small molecules. And we can address the key issues in peptide drug discoveries. And these include finding the basic sequence, improving the in vitro activity, enhancing in vitro stability, and extending in vivo duration, and even achieving the oral absorption, which are challenging for peptides. And our comprehensive solution means that we can address all peptide drug discovery challenges in one place. This allows us to not just make peptides, but to make peptides that can become real medicines. Let me show you some case studies of our peptide drug discovery results. First one is an NMUR2 agonist, a potent and highly selective candidate peptide. And you can see the chemical structure here, showing how we optimize both activity and selectivity through careful design. As a result of that, we have successfully identified a candidate peptide with in vivo efficacy. Second one in the center is a GIP agonist, a potent and extremely long-acting peptide. In this project, we created an optimal duration peptide by chemical modifications. When we change the alkyl moiety length, we can arrange the PK profile. As a result, we have found 1 week acting peptides. And third one is a peptide drug conjugate. This shows how we can combine the peptide technology with conjugation technologies. In this project, we successfully identified the combined molecule of a cyclic peptide and oligonucleotide with linker. These examples show that we can handle different type of peptide challenges, from selectivity, duration to the novel conjugation approaches. Each project requires a different expertise, but our scientists and our integrated platform allowed us to address all these different needs. The publication and patent application listed here demonstrates that our work meets the highest scientist standard and create a valuable intellectual properties. Next case study about peptide synthesis. Axcelead peptide capability enables a rapid synthesis using a bunch of automated synthesizer and purification systems. Furthermore, we can access to the highly complex compounds, including bicyclic and tricyclic and fatty acid modified peptide and so on. And as shown in the left side, we synthesize more than 1,000 complicated cyclic peptides per year, including that kind of a peptide. Our success rate of synthesis is now more than 95%. Next case study is MK0616 analog. If you are a peptide expert, you know that this peptide is one of the most difficult to synthesize among clinical peptides. Despite this situation, we completed the synthesis within 1 month, which is very fast for the complex peptides. This speed comes from not only our combination of automated synthesizer and purification systems, but also our synthetic expertise that combine both peptide and small molecules. The other case study is oligonucleotide. In this study, we've constructed a robust synthetic route and efficient purification methods. Lastly, I want to introduce you our design technology using our priority insert tools. Let me show you how this works using a case study of a PC case 9. As shown here, we have three main computational tools. First is the amino acid residue by residue interaction analysis, and this helps us understand which part of the peptide are most important for binding to the target. And second is the spot exploration and fragment drug design tools. This gives us information about the binding fragment that we can use for structure-based drug design. And third one is automated proposal tool for new structures. This can generate new structures for specific substructures using ligand-based approaches. In these studies, we can identify the important interaction and then design the novel structure to improve these interactions. In summary, for the peptide, we can provide you high-value peptide drug discovery by the combined peptide platform with advanced technologies developed at Axcelead. Okay, now let's move to the oligonucleotides. This is another very important new modality that can address the genetic target directly. For this modality, Axcelead enables the creation of a potent and safer candidates through our one-stop drug discovery service for oligonucleotide drugs. And as shown here, we have two key strengths in oligonucleotide discovery. First is our robust platform from discovery to IND. We can design with our original algorithm and evaluate efficacy and safety using knock-in animals and provide a full set of in vitro and in vivo toxicity and DMPK assessment. Second is our solution with various technologies. We work with a novel chemical modified oligonucleotide with MC amidite powered by Nissan Chemical Corporation. And we can visualize the oligonucleotide distribution for PK/PD analysis. And also we can provide a targeted delivery with peptide oligonucleotide conjugates. Let me show you a case study of a sequence optimization of a PNPLA3 gapmer ACE role. In this study, our goal was to identify the candidates with a good balance between the activity and safety. This is always a key challenge with oligonucleotide. The graph at the bottom shows the different oligonucleotide design and their effects on the target messenger RNA expression and cytotoxicity. In this study, the lead sequence on the left was 16 RNA, and then by extending it on 18 and converting it to the MOE, we were able to improve the knockdown activity. And RNA MOE mix and RNA matrix mix were also improved the knockdown potency, but cytotoxicity went to the bad directions. In this study, finally, the best result in the circle were obtained when some of the PS bonds in the wing region were replaced by the PO bonds. This demonstrated our one-stop drug discovery approach with design, synthesis, and evaluation for both activity and safety. Okay, now let's look at the third modality drug conjugate. This modality combines the targeting ability of a larger molecule with the potent activity of drugs with linkers. Axcelead drug conjugate capabilities enable the development of antibody drug conjugate ADC as well as peptide drug conjugate PDCs. We can synthesize the highly complex and various compounds with different linker, payload, and delivery component. The chemical structure shown here demonstrating the complexity conjugate compound. We can work with RGD peptide for targeting in green and various drug payload in red and cleavable and non-cleavable linker in blue that controlling the drug release. Each compound have a different properties, but we can synthesize such compounds because we have expertise in multiple modalities. Our comprehensive and integrated capabilities enable the optimization of all these moieties together. Next, the fourth modality is a targeted protein degraders. We support both bifunctional degraders and molecular glues. And also we have a command degrader platform. Our platform covers the complete process from target identification to the ID enabling studies. For bifunctional degraders, we have high throughput synthesis and purification capabilities, a degrader chemical toolbox, and ADME solution to discover the oral drugs. And for molecular glue degraders, we focus on the neo substrate identification and molecular glue focus library, high throughput screening, and mechanism of action identification. In case of TPD, this comprehensive approach is necessary because protein degradation is a complex biological process that requires understanding of many different factors. Let me show you our specific capability for the bifunctional degrader synthesis. As shown here, we can provide rapid access to the 96 targeted protein degraders in just 1 month through multi-step high-throughput synthesis. This speed advantage allows for rapid optimization cycles. In addition, our degrader chemical toolbox opens a new chemical space. We have more than 500 protein of interest binders and more than 50 E3 binders and thousands of different linkers. This toolbox approaches mean that we can rapidly explore the many different combinations to find the optimal degraders for each target without building block synthesis. Finally, let's look at the RNA targeted small molecules. Our approach has 4 main components. First is our RNA focus library, which includes both RNA splicing focus compound and general RNA focus compounds selected. And second, in the top right corner is our screening and profiling capabilities, including the various receptor, various reporter and binding assays, and selectively profiling with transcriptomics. Third, in the bottom left is our compound evaluation capabilities, covering pharmacology, DMPK, and safety from target ID to ID enabling studies. And fourth, in the bottom right is our drug design and synthesis capabilities, including RNA-focused high-throughput synthesis, machine learning methods for RNA binder prediction, and ADME-tox prediction models. These capabilities allow us to discover novel RNA-targeted small molecule. And let me give you more detail about our RNA-focused small molecule libraries. As shown here, we have 3 different libraries. First one is our RNA splicing focus library with approximately 2,500 newly designed and synthesized compounds. This is the only library of its kind, and all compounds are likely novel with a potential intellectual property value. We have track record of hit discovery with extremely high hit rates for various targets. And second and third general RNA focus library with 6,400 compounds selected from our 1.2 million compound libraries. And these were selected through multiple analysis of various RNA binders and followed by the prioritization based on the RNA binder-like properties and drug-like properties. These libraries show the impressive track record with more than 10-fold higher hit rate than our diverse library. Lastly, one key challenge of this modality is designing a compound without admitox issues. We try to solve these issues by constructing of more than 15 probability admitox prediction models. Our approach uses manually curated training data for machine learning and utilize our large admitox database originating from pharmaceutical companies. This database contains approximately 150,000 data points for accuracy, solubility, and reproficiency, and so on. We use automated machine learning with rigorous validation. This comprehensive approach allows us to predict the admitox properties early in the design process, which help us to design the better compounds from the beginning of the drug discovery, rather than discovering a problem later in development. The combination of pharmaceutical quality data with recent advanced machine learning gives us a very useful prediction capabilities that's better than the commercial available models. Now let me summarize what we have covered today. And as a summary, at Axcelead, we would like to realize a world with no borders between modalities. To realize this vision, we offer three key elements. First, Axcelead delivers a pharma standard integrated drug discovery platform. We combine the people, technology, and data to address client needs. Second one, we provide solution to the challenges for new chemical modalities, advanced chemistry for candidate generation, and total and seamless evaluation system, and diverse approaches to the modality selection. Third, we provide the technologies in each chemical modalities, peptide, oligonucleotide, drug conjugate, targeted protein degraders, and RNA targeted small molecules. We'd like to work together with clients to find the best solution for drug discovery of new chemical modalities. Thank you very much for your attention. This is the end of my talk.
Thank you very much, Yoshito, for an excellent session so far. Before we begin today's Q&A, let's run through some brief announcements. First, I'd like to thank Axcelead Drug Discovery Partners Incorporated for sponsoring today's session. And while we already have a few questions in the queue, if you have a question for Yoshito, please make sure you submit it now in the Q&A module on the left-hand side of your screen. And with that, Yoshito, let's go back to you for the Q&A. All right, Yoshito, thank you. Our first question here, for peptide synthesis, how many building blocks of amino acids and unnatural amino acids are in hand in Axcelead?
Okay. Thank you for the question. Now we have greater than 500 building blocks of amino acid and natural amino acid in stock. And we can synthesize many kinds of peptides from linear to complex peptides very quickly by using these building blocks and five automated synthesizer. Yes, we have a bunch of amino acid building blocks in stock. This is my answer.
Perfect, thank you. Our next question here, for oligonucleotide evaluation, can you conduct in vitro, in vivo testing and safety study in Axcelead?
Okay, yes, we can conduct all nucleotide assay in Axcelead. For example, we can evaluate knockdown activity and cell toxicity assay in vitro. And for in vivo assay, we can evaluate efficacy and safety by using knock-in mouse. because we can produce a knock-in mouse internally. And in addition, we can visualize the oligonucleotide distribution for PK/PD analysis. Thank you.
Great. Thank you, Yoshito. Another question here. For drug conjugate, what types of conjugate modalities are available in Axcelead?
Okay, thank you for the questions. Now we are available for the drug discovery of ADC, antibody drug conjugate, and AOC, antibody oligo conjugate. And also we can approach to the PDC, peptide drug conjugate, and POC, peptide oligonucleotide conjugate. And because we have the capability of a small molecule peptide and oligonucleotide capability, so we can access to the all these modalities. Thank you.
All right. We have two more questions here, Yoshito.
Mm.
Next one is, what are Axcelead's competitive advantages compared to other CROs?
Okay, this is a very important question for us. I think we have three main differentiation points. And first one is most of our scientists were coming from pharmaceutical company, and average career is more than 15 years. Uh, Therefore, we can understand your needs as a drug discovery, and we deliver the highest standard scientists. And second one is we have a 1.2 million compound library and 1,000 project data directly inherited from pharmaceutical companies. And we can utilize the compounds and data for accelerating the project for our client. And the third one is we conduct the continuously research across the chemistry, screening, DMPK, safety, and pharmacologies, because all functions are in the same building. This speed advantage means faster decision making, more efficient use of resources, and ultimately faster progression towards clinical candidates.
All right. Thank you, Yoshito. And our last question today. What types of services does Axcelead provide, such as FFS or FTE?
Okay, thank you for the questions. And we can say that depending on the client needs, we are very flexible. And in the case of chemistry, currently we have FT services and FIFO services and integrated drug discovery with milestone setting. Again, we can say we are very flexible for contract. And if you have interest in our drug discovery service, please contact us from our website or something. Thank you.
And that's all the time we have today. Yoshito, thank you very much for an excellent session. If anyone submitted a question that wasn't addressed, keep in mind that the speaker will reach out to you directly. This session was recorded. You'll receive a notification in 24 hours when the on-demand session is available for viewing. And before you log off, please take a moment to complete the feedback form so we can continue to improve your webcast experience. On behalf of BioXeconomy, thank you and have a great day.